Hello, thank you for warm welcome. Okay, a couple of words about me. I am a neuroscientist. I have a PhD in psychology, surprise, but in psychophysiology. And I'm a cognitive neuroscientist, which means that I do not cut anything, any living creature apart. Okay, so today we're gonna talk about AI in neuroscience. To be honest, because these two guys are closely related to each other. Some of AI and of course artificial neural networks were inspired by neuroscience, and vice versa. AI now affects neuroscience as well, and that's good. And we're gonna browse. We're gonna go through this small presentation to understand and see where these methods could be applied in mostly cognitive neuroscience. Why so? Because if we would go to the cellular level, to the biochemistry and so on, it will take kind of one day to cover everything. Okay then, first of all, just to mention it, kind of pre-AI methods, so ICA, PCA, CSP, and ISC. These guys are kind of more or less commonly used in neuroscience. Why and for what? ICA and PCA, mostly we need, okay, do you recognize this course? I think maybe not. This is electroencephalography. So each curve explains and shows you the electric potential from a given point on the head. So I believe that you saw this method, or maybe even suffered from this method in the past. Basically, your brain, your cortex, and the organization of the neurons in the cortex allow the electrical field to spread over the scalp, and we can collect this information. But the problem is we are deeply electrical creatures, so everything is charged. Our tongue is charged, negatively of course. Our eyes are charged, and so on and so forth. So we have a lot of artifacts around us. For example, in the center you can see that IC decomposed the signal, and we may see different components plotted there. So we can see blinks, the muscle artifacts. You see that the frequency of the signal is much higher, so they are kind of hairy. We have some noise, again, from the sockets and everything around us. So basically, this poor EEG approach suffers much from everything around and in us. The other part of the EEG, kind of counterpart of the EEG, is MG. It's magnetic electroencephalography, so it gathers the information about the magnetic changes, and basically it looks quite the same. So ICA or PCA could be used to clean our data, first of all. And of course we need some number of electrodes. Maybe you saw this brand new kind of wearable devices, kind of BCI, brain computer interfaces, or BFA feedback that is spread over a kind of market. And usually they have like 5-6 electrodes, and you can just buy it and play with it. But this number of electrodes is, of course, is not that good to make the composition effectively, and so on. Usually we have kind of, here we have kind of 20 electrodes, and usually we can gather information from like 64 or more than 100 electrodes. So these guys help us to understand where we have the raw signal and where we have the kind of brain signals. And this is a common tool to use in EEG, MEG, and other approaches. Another tool here, the same tool, but another approach here is to use PCA and RCA to decompose signal that related to some event. So this disturbance there is caused by, for example, a sound or a visual stimuli or something like this. And each deflection of each curve, basically red, blue, or black, is a component. In some cases, we need to isolate these components from each other, just to understand. For example, if you want to work with the brain-computer interfaces, we can train our BCI to catch this component, and the easiest way, kind of before any training, is to use PCA or ICA approaches to isolate one of them and focus all our attention on this amplitude or whatever. The third one, CSP or common spatial pattern. The point is of this method, again, it's commonly used in brain-computer interfaces to separate different sources of the signal. Of course, all of these, this is topography, so the colors depict the distribution of electrical field. In this case, these are the CSP weights, but the same picture you will see in EEG or MEG. The point is, all of these different sources are projected together on the scalp, so they are mixed together, and CSP helps us to separate them. And next step, if we need a person to control some robotic hand or whatever, we can focus our attention on some part of electrodes and use this signal as a signal that helps a person to manipulate a computer, for example. Okay, ISC, intersubject correlations. Again, it's not the II itself, but ISC is typically used to understand whether a group of people was watching some movie or some video attentively or not. The point is, now you're listening to me, and some part of your brain activity is quite similar to each other. You hear the same, you see pretty much the same, and of course in case of a movie you see basically the same. And this idea came from fMRI, functional magnetic resonance imaging. So MRI, I think you know what it is. And fMRI is looking not at the brain structure, but at the changes in the blood oxygen level. So basically, fMRI looks how your brain eats, basically, breathe, to be precise. Okay, and this bold is for blood oxygenation level dependent signal. So these curves reflect each voxel, which is a 3D pixel of this model, and the changes in the blood flow in there. And the logic is quite simple. If we have the same pattern of activity across people, the blood flow will change kind of alike in some part. And we can maximize this function, this correlation, by applying weights onto our voxels. And so therefore we can predict and we can understand if some person were listening or looking attentively, or in other case, which part of the video was perceived and remembered. So basically this data predicts your recall of the movie and so on and so forth. And this method, again, it started from fMRI, then it came to EEG, I mentioned before, then it went to eye tracking, Do you know what it is? Okay. Okay. Eye tracking is a very simple method that uses a camera. In the professional way, it will be an infrared camera to understand where do you look. So it can track my gaze. And again, if we look and if we watch the same video, the pattern of eye movement should be quite the same. And again, this method, very simplified, showed that these scores of correlation predict the points that people gathered after this kind of lecture or educative video. Okay, then we should go to BCI again, and BCI brain computer interface is the huge family of everything basically. It could use any type of input, even PET, positron emission tomography. In this case, basically you need to provide your subject with some radioactive materials. But for BCI, for daily use, it's not that useful. But anyway, and of course, each person, each research group in using any of this method tries to apply some kind of AI to teach their model to be more effective, to be more fast, to be as effective and as precise as possible. But again, now we have kind of 25 years from the BCI 2000, and no big leap forward. But we have something in the invasive techniques. Here we can see a grid of electrodes that's been plugged into the living brain, and the diagram on the left upper corner, you may see this that each black point there is a spike of a neuron, so the neuron fires there. What did they try? They tried to teach a person to speak by kind of power of the brain. It's pompous, but something like this. And they tried to use a language model that predicts not the letter but the phoneme and help a person to speak. Of course, this is not a kind of a healthy person, first of all. And basically they succeeded. So the word frequency levels of these people, their subject, were able to speak at the pace of kind of 62 words per minute. It's not that big. My tempo is kind of 160, I believe, right now. But comparing to other methods, it's a big advance. And again, and I know that almost of you heard about the Neuralink by Elon Musk, and of course they use approaches that are more or less close to this depicted here. And the next stop here is AI and neuroimaging. Neuroimaging is kind of family of methods that helps us to understand what's going on in the brain from the perspective of structure or function, and therefore we may use, and people are trying to use AI methods, all of the family of these methods, to somehow predict, for example, in some cases, predict what does the person see. In some cases, try to use these approaches to manipulate something, again, BCI. In some cases, to help medical personnel to navigate through MRI, for example, if in many hospitals around the world, you cannot find an MRI a device with high quality, kind of 5 Tesla or something like this. So you may use AI that allow you to artificially enhance the resolution, and therefore the probability of correct diagnosis rises up. And of course, next step here. And of course, we'd like to understand what do the people see, and we would like to understand and predict what they think about. The second point is it's almost impossible to predict because we have a huge amount of thoughts in our brain and at the same time. But if we focus on the visual cortex, it's right here, and then train our model to understand which voxel, which part of the functional imaging predict some specific pixel here. Some of the research groups were quite successful in the reconstruction. You may see on the left it's a visual input, and here we see the reconstruction. Of course, it's the best result, and it will take some time. But in any case, I think it's again a step forward. But again, the same approach, and again, even more precision here. And here we compare different approaches. And we'll return to this one, to Miyawaki, a bit later. What else? And if we'll try to merge these approaches with the generative AI, we could see something like this. So here we have stimuli and the predictions. So I believe here is the stimulus, and these are the predictions. It's not kind of predictions, it's kind of the results of generative model working. Not the best, but I think it's again something interesting. I'm not sure what will go. Okay, I think in 10 years maybe we'll see something even more kind of peculiar. And I hope that nobody will be able to watch my dreams, to be honest. But in any case, these approaches developing really fast. And for example, if you want to work with some, if you are medical personnel, this is quite useful. So on the left side, you may see the structural MRIs. Oh, I'm lying. It's computed tomography. Basically, you can see that these are the raw pictures of the CT, and here is, we may see that some of the points here, some of the areas here, are highlighted by the AI, and you may see there the principles of this. You may see here an area, and on the right picture there you can see the kind of whitish part, and this is about the ischemic stroke. So it's not that easy for personnel, or for example, in some cases you can't approach a doctor. Doctor could be kind of busy or basically absent in this specific place, but kind of a nurse or some kind of, or kind of low-level medical personnel can use these types of analysis to pre-diagnose the person. So this one should go to the hospital as fast as possible, of course. And of course, if we have this peculiar, not very understandable thing that called AI, I believe that This is a big opportunity to diagnose everything, basically. You may see on the left, this is a picture from the big review, and they were trying to understand which specific pathologies and so on and so far were tried kind of to diagnose with AI, some type of AI. And you may see here that basically everything, any brain injury, epilepsy, sclerosis, Parkinson's disease, brain lesion, schizophrenia, Alzheimer, brain tumor, whatever, and of course the tumors are the most kind of loved here, since they have a specific structural pattern. And again, how can we do it? Of course, with MRI first, but also we may use the functional connectivity, which is about the links in functional changes between different regions of the brain. For example, all our visual system is linked together. So if we see some activity on the retina, for sure you will soon see this activity in the visual area 1. And so on. And some of the approaches allow you to build kind of huge tables of interconnections of different regions. And these, for example, coherency scores may be used as features to train the model and return the results. For example, in some cases, you can even guess the age of a person by the resting state brain activity. Okay then. Also, okay, functional connectivity. There is SC is for structural connectivity, so we can see the wires between the different parts of the cortex. PET, I described it before. CT, computed tomography, and DTI is basically it's a diffusion tensor imaging. It's used to localize and understand the firmness of the white matter in the brain. So white matter is kind of a bunch of axons, so they are kind of wires, and we can understand the spatial and kind of vector of these tracts, and use this information to predict something and to understand whether it's a healthy person or a person with a disease. Or we may try to do this. Basically, none of these, to be honest, are used as a kind of medical standard. No. But they approach it and approach it. A lot of studies about it. Okay, and so these are the methods of AI. You may see that basically almost everything was tried here. So AdaBoost, RAF, and so on and so forth. So basically people are trying to use each of these approaches to somehow predict, to be more precise, to use different type of regularizations and so on and so forth to get this big fruit. Since if you will be able to make early diagnosis, for example, for Alzheimer, and it will work, and it will be working for 10 years with high robust, you'll get the Nobel Prize, I believe, because this is a really big, this is really important. I found the word. Okay, what else? And it's not kind of neuroscience itself, but it's kind of nice. Why not to model human behavior by brain activity or by previous behavior or by condition or whatever? Try to use a model understanding child behavior. Sometimes the child behavior is something strange, and if you need to understand, you just go to some university and just leave your child there, and in 4 or 5 days maybe you'll receive some matrix with weights, and possibly it will help you. Also, again, we can model traveling of students using, I don't know why, but why not? And here is an example of the of a CT image. And on the left, basically, you can see, or maybe not, or maybe yes, some changes in the tissue. But here, AI can help you to find these changes, these differences from the healthy brain picture. Okay, this is the first part, and now we have kind of, we have kind of 7 minutes to go. And the toolbox that I decided to show today is NeLearn. You may easily find it. It's free, completely free, and it has good tutorials. And this guy is focused on the fMRI and data analysis. And today I decided today to use their examples of how to use it, just because my data is kind of boring, to be honest. I don't like to show you kind of tables and then describe each of the number and so on. You'll be sleeping in 5 minutes. Okay, then, now I quit this. Hop. Now I go here. I told you that we will return to the Miyawaki, and this is kind of an old story, to be honest. Also, you can find and download and play with this script as well. Again, I can give you the link to this to their site, of course. And they were trying to predict specific picture based on the mask of voxels, mask of fMRI activity placed on the visual fields. And this is a big work, to be honest. Each brain is different, and you need to take this brain, then you need to to make a model, of course, the 3D model. Then you need to pick the voxels that are connected to some visual area. Then you need to decode this, so it's not a 3D image now. It's a vector, and then you'll be trying to do to move forward. So they used fMRI signals from a kind of simple experiment. Basically, they had this 10 by 10 matrix that could change the color from white to black, so that's a grayscale. And then they tried to teach the model to reproduce, it feels like I speak too loud, to reproduce the image that a person sees. So they used not only kind of decoding of each voxel, each pixel by one, but also they were focused on the contrasts between the neighboring pixels. So you can see here that in the middle, 1 by 2 and 2 by 1, these kind of wide and long matrices, they give more information about the contrast of the neighboring pixels. Okay, here we need to import something and load the data set, which is not that big, kind of maybe 10 gigabytes. And here we are, okay, I see. In any case, the most important thing is there. First of all, we need to use the mask. And this dataset contains the initial masks, and the library itself has a bunch of functions that allow you to decode. They are called decoders. And they allow you to approach these specific voxels from the model and simply make a table from there. So it's not a huge image with different parameters. It's just a vector that we could use to train the model. Okay, I didn't expect this, to be honest. It should have been made in white. I believe I can change the theme. Yes, I think yes, I can. And now you will see me suffering and in trying to find hope. Wait a second, appearance. It should have themes. Am I right? In any case, okay, I'll try. One more. Okay, let's go here. Nothing. Why I do not like IDEs is the number of options they have. You can find anything but what? What else? Let's find preference. Wait. I'm too old. I'm old and it should be somewhere here. It should be appearance, yes, but yeah, yes, appearance, and then color scheme. Okay. Okay. It's accumulating both for me and for you. Now you can see somehow what's going on here. In any case, it's not something very understandable and very simple to get, but the reason is we just need to iteratively go through the model and keep our voxels and transform them into this vector I was talking about. And then we define the prediction function that trains, that use orthogonal matching pursuit to train and to find the weights that will maximize the correlation between the intensity of some pixel and activity of a bunch of voxels. So it doesn't take a lot of time for this data set, but okay, then we run this prediction, and the nice point here is we can see what we have here. So basically these pictures on the left, they are ground truth, and then we have a reconstruction that we could binarize to see it with kind of black and white representation. And you may see that if we use these masks correctly, basically we can, and this is real data, to be honest, and we can predict what did the person see, which is quite nice. And to be honest, this is, okay, I go back here and I want to show you this. And this is the full results of their study, kind of. You may see, and each of these strings, each of these images is a single trial that took kind of 12 seconds of flickering of this letter or figure, whatever. And you may see that in some cases, for some subjects, you may see the better picture. For some subjects, you may see the kind of not that good picture. But it was kind of 15 years ago. And now we can reconstruct it with much better resolution. But the point is, in many cases, these approaches, they are kind of demonstration of technology, mostly. For example, if you want to understand how does the brain work in the case you see a cat, or in the case you see a face, or in the case you see a chair, you do not need the AI approaches and long training and big training sets, and then again big validation sets to do this. Basically, you usually use very simple approaches and parametrical statistics, and it's enough to separate them and to understand which parts of the free-form gyres that it's in there is responsible for phase, for house, and or whatever. But step by step, all of these approaches in BCI, in EEG, in MG, help to model and the brain activity kind of in more conceptual way. The problem is that each data set with neuroimaging, whether it's MG, EEG, fMRI, whatever, it contains a lot of data. And the first of the problems is we suffer very much when we do statistics, we suffer very much from the correction for multiple comparisons, because we have the tens of thousands of comparisons, basically. And we have this system that is very, very complicated, and it's very, it's very hard to formalize all of the relationships between kind of voxels and so on. So it looks like data science and big data approaches will be quite useful and will be very, very effective in this case. But the problem is, if we need a lot of data, we need to somehow collect them from different universities, and they sometimes are kind of greedy. And we need to collect this data from different research groups, and they have their own standards, and so on. So we have kind of a huge bunch of data. But to standardize it, you need to spend kind of years just to collect all the data. And in many research like this, there are not a lot of people, not a lot of subjects. So it's kind of 60 subjects for train and 60 to validate, something like this. It could be, this is kind of order of number. But if you need thousands, and basically we have these thousands, because a lot of data is stored across the globe in each MRI department of each university and medical center. But in this case, we need to collaborate, and I think this is a big point of big data science in neurobiology, to be honest. But again, here is the place when maybe I'll stop, because the time pass. And thank you for your attention. I hope that you've learned something new here. I hope that it was more or less interesting for you. I'm not sure if it was, because it's kind of different angle and kind of step aside from the main course of PyData events. But again, this was Aleksei Gorin, and thank you for your attention.
Thank you, Aleksei. My name is Ilya. Thank you very much for the lecture. It was certainly very interesting for me. I wanted to ask you, do you know maybe of any particular examples of how machine learning models or any kind of AI models were successfully or relatively successfully applied in real, like, clinical practice, maybe for diagnosis?
The point is, medical care in each country is something that is very hard to get in.
It's very conservative.
Yes,
yeah.
It's very conservative. Even if you have something like a very simple, a new pill, it could take 10 years to put this pill in the insurance list. And again, if the main question here is quite simple. Two questions are here. First, what if AI will be wrong? What about the kind of misdiagnosis? who will be responsible? A doctor, a company, a hospital, or who? And the second point is, even this guy is so effective, what's about my job? So it's, I think in some cases or in some countries with a kind of more more alive regulators, it could be used in some cases, but I don't know about. And okay, some types of software could be purchased by medical centers, but it is not a standard. It's their own decision and their own risks. Okay. So in this case, the only thing that could be quite useful and only software that is very useful for medical personnel is the tool that I told about before, sorry, that allow you to boost up the resolution of MRI. And I think that they will be successful in this part. But if the AI tries to diagnose something, that now we have a human factor and bureaucracy factor here. Too much responsibility, too many people are too conservative, and it works against AI interventions in medical sphere.
Thank you. Thanks for presentation. If I do understand correctly, that neuroimaging is giving us opportunity to visualize what kind of parts in the brain are being activated. For example, when we are watching something or we are listening. And I just wanted to know, is it giving like precise information which neurons are being activated, and maybe AI helping us to get that information or predict that exactly these neurons among, for example, 10 or 100 people are being activated when we are watching some specific thing or specific picture, for example?
This is a good question, and I thank you very much for it. And the point is that the resolution of fMRI, for example, that gives us the best 3D spaced information about the neuron. First point, it looks at the blood level changes, and they are not immediate. Neuron fires at 1 kilohertz. Bold activity, these changes in blood flow took like 6 seconds. Kind of 6,000 of events could happen during this period. So this approach will, and I believe that these approaches are involved in the field of the wet biology, when you plug some electrodes into the brain of an animal or of a man, and it could boost up the resolution of this electrode grid. But and the second problem of fMRI, for example, that is that voxel, this elementary part of this 3D picture, is maybe 3 by 3 millimeters, and in this volume we have thousands of neurons, so we can't focus this good. And basically, in any case of non-invasive neuroimaging, we work with the neuronal populations mostly. So we work with again thousands, tens of thousands neurons. But where the AI could be very useful is in the non-brain, not in brain imaging, but in brain stimulation. Because if we apply some electricity, very tiny currents on the scalp, it could affect slightly the brain. So it's nothing like electroconvulsive therapy, okay? And these changes could be seen by neuroimaging and also by changes in behavior, for example, changes in reaction time. But the problem is, again, the brains are very different, and the currents go through our head with big distortions. So it's not a sphere of copper. we have a bone, we have bones, we have gray matter, white matter, whatever. And the currents go in a really peculiar ways, could go, and you cannot simply take these electrons from one person and put on another and be sure that the region that is stimulated is quite the same than previously. And in this case, to model it, it takes kind of 3 hours and something in some cases. And here is where AI, I believe, could help. So you can model these currents based on the previous teaching of a model much faster than the formally calculate this.
Thank you.
Yeah, thanks for the lecture. There are some well-known issues which arise when solving AI problems, like data leakage, overfitting, or unbalanced data. Maybe there are some problems which are highly specific for neuroscience.
Okay, there are problems that's specific for neuroscience, and one of them is, for example, our brain works in not on a linear scale. We usually you need to use a lot of regularizations, and the results of. Okay, let me give you an example. There was a nice project where people, the researchers, were given with the same, exactly the same data set. And they ran their analysis, quite the same. It should be the same, of course. But the slight changes in their approaches basically flipped the results. So the distribution of the results was so big. So, the problem here is the human factor also, because all of pre-processing, all of the data collection part and so on, each of these little parts could change the results dramatically. So, for example, if we will try to collect the data, but again, I went from the question, of course. The problem here is that usually just do not have enough data. That's the first problem in some cases.
Okay, but if you don't have enough data, how can you run some very complicated AI methods? The less data you have, the more robust methods you need to use. Like, it's more about statistics, not about some complicated AI stuff. No neural networks.
Yep. I'm telling about, for example, this type of prediction is more or less raw, so it's not that precise. If you want to make the better predictions and so on, you need to have more and more people in your sample, and basically it's too expensive to get this much data, and this is why people around the world are trying to collaborate and gather as much as possible standardized data to work together here. Also, another approach is kind of, if we have not enough data, how we can kind of make it more precise using AI approaches. But again, I'm not really familiar with this type of research. And also many researchers who are trying to apply ML somehow in their studies. Basically, they are not mathematicians, they are not kind of software developers, and in many cases, these applications are very naive. So in some cases, for example, I saw a case where people reported kind of 80% or 90% prediction score, but they trained and evaluated their model on the same bunch of data. So it shouldn't work like that. So basically, we need a kind of, again, we need to collaborate. We need to have really high-level physicist or mathematician or data scientist in the team. And also we should have a psychologist usually, a biologist who understands how the brain works, and they should somehow work together. And it's almost a fairy tale. It's usually it doesn't work like this.
Okay, thank you very much.
Aleksei, I agree completely with everything you have just said, but I wanted just to add a little bit to your answer, because I work, I think, in a pretty similar sphere, and this issue is very important for me. So indeed, we have a very serious problem of small data sets, and also we have very bad signal-to-noise ratio, and also usually a lot of our features we use are very hard to interpret, so it complicates feature engineering in many cases. And also a very harsh problem of multicollinearity. So we have to deal with all of this at once. And one of the best approaches we found in our practice is basically to double down on cross-validation. So techniques like nested cross-validation. And the same approaches which work in industry where you can always gather some additional data, they do not work as well here, because usually you have some fixed data set, and this is what you have for years to come. So you have to get most of it basically by applying additional computational resources.
Can you name some typical go-to models you use in your day-to-day tasks?
In our case, we use the simplest models, logistic regression. Also because, yeah.
That's a nice model.
It is sufficient in our cases because we also try other types of models, more complex ones, but they usually do not outperform the simplest ones. And also in medicine, it is crucial to have interpretability and clarity of the model, thus linear models.
Thank you very much for your lecture. I have two, questions. The first one, am I right that in order to get some data which could be meaningful for those algorithms, you basically need to put a huge device at somebody's head and to record some data for hours?
In some cases, you need to put subject's head into the barrel full of liquid helium, to be honest. So in some cases, you need to put somebody in the place that sounds like a techno party, which is MRI. Yes, we need the problem of neurobiology, a big problem of neurobiology itself, the cognitive neurobiology is it's not ecologically valid at all. So everything we have basically usually be collected in the laboratory in the environment that completely unnatural for a person. And hours, maybe kind of hour and a half, maybe that.
So how do you see perspectives on solving some tasks from regular life?
Which kind of tasks?
I don't know, just like...
Oh, if we are maybe in the medical part of this, okay, that's the problem of subject that he needs to lie down for an hour in the MRI device. It's he or she do not have that much choices. In the case of the wearable devices, there are some approaches. First of all, I should mention that another method, functional near-infrared spectroscopy, is becoming more and more robust, I believe, and more compact. So the point is that our heads and we are kind of semi-transparent for the near-infrared spectrum. So we are like big bubbles with the snow. So you can use a laser, and this laser can penetrate your skull, not physically, okay? The light goes inside, and also it scatters and returns back here. So you place a light source here, and close by you put a sensor. And again, if I took this glass and fill it with the blood, you can guess whether it took from a vein or an artery, right? And these color differences are even more pronounced in the near-infrared spectrum. So we can use two lasers that will give our information. Will give our information about the same blood oxygen levels in this tissue. Problem is with the spatial resolution, of course. And there's another problem is that you cannot go too deep. So basically, you take only information about the higher about the cortex. You can't go deep inside for now. And these devices now could be very compact and wearable, and also combined with EEG. So I saw an experiment where a person was put into a helmet with the EEG sensors together with FNIRS optical sensors, and with glasses with a video camera that also had a built-in eye tracker, and the person was simply walking around and looking at everything. And so the AI that was built into the camera classified the objects where the eye tracking was pointed on. So like if I look here, the duck, okay? And simultaneously an event drops into the data of FNIRS and EEG, and people are trying to somehow use this data to understand which type of object is in front of a person by using this FNIRS EEG combined approach. I'm not sure if it's an effect of something, but in any case.
Thank you.
Hello. Thank you for your talk. Are those results from the, did you say the visual cortex?
Yeah, these are results from the visual V1, the primary visual cortex.
Okay. It's very interesting. I'm not a neuro person, but a part of me wonders, like, when the brain sees something, it's not purely a visual phenomenon. If I see a chair in the middle of the room, I also like the concept of a chair is triggered in my head, and I can imagine a chair even without seeing it. So I don't know. I guess part of me wonders if this is accounted for. Can be accounted for.
Yes, it's. I didn't mention it because it would take some time. The problem is you can see that we have a white on black pictures with a very high contrast, and so on. And they use these masks on the primary, secondary visual cortex and the V3 again, as well. Sorry. Because these regions kind of work with the very simple features of the image. In this case, we do not have any concept, so we have a row of pixels and very simple geometric shapes. Of course, our visual system works in many steps. And for example, if you want to talk about chairs, there is again a research that uses information from different masks. So our brain sends from the visual cortex. You could go in two ways. So one is dorsal, one is ventral. So here we know the dynamics of the environment by these parts, and by the parts that are kind of under the temporal part of the brain. we classify the objects into different categories, and this concept of chair lives there, and if we put the mask there, we could understand and we could classify. We cannot, I believe, can't for now recognize which type of chair do you see in front of you, but we can understand if it was a chair or a house or a car, kind of 5 classes, and it's possible to recognize which kind of classes is in front of you.
Thank you.
I'm interested most about your research, and also is there any other kind of opportunities in Armenia to do this kind of research centers?
Basically, currently I am kind of a neuroscientist on the way, so I have some research that are currently not finished, and now I'm working as a kind of R&D or data analyst in a startup. But if you're interested in some projects from the perspective of data analysis or data science or whatever, I'll be happy to help you, so you can approach me and we can share the contacts. That's it.
Thank you.